Okay, so again, you've seen these pictures before. These musculoskeletal receptors are located in muscles and tendons and joints. So we're going to start with the muscle spindles. So again, the intrafusal fibers run parallel to the extrafusal fibers. They monitor changes in muscle length and velocity of that change. Their vital role is measuring position, movement sense, and they help with motor learning. So the type 1A afferents that you can see on this left picture, they monitor quick stretch. So these are in fact phasic, which means they quickly respond to the quick stretch and then they stop responding. And then the type 2 fibers discharge during constant stretch. So they're tonic in nature, so they're constantly firing. So the 1A afferents that monitor the quick stretch are in the center of the nuclear chain bag and the nuclear bag fibers. And you'll notice that the 2s are a little further out, a little closer to the endings. So when we have the small efferent fibers at the end of those type 2 afferents, at the end of the muscle spindle, they adjust the spindle fiber stretch so that the spindle can be responsive throughout the physiological range of muscle length, because we want the spindle to be constantly firing. So to maintain sensitivity of the spindle throughout the normal range, those gamma motor neurons are firing, causing the end of the intrafusal fibers to contract. And that contracts at the ends of the intrafusal fibers, and it stretches that central region of the spindle because, and there it maintains that sensory activity of the spindle. So again, you need to remember that the intrafusal fiber ends are attached to the extrafusal fiber ends. So when we stretch the skeletal muscle, that extrafusal fibers, when they stretch, it also stretches the intrafusal fibers, which stretches that central region, because the intrafusal fibers are contractile only at their ends, not that central region. So we also have the Golgi tendon organs, which I know you've probably seen a bunch of times. So these are encapsulated nerve endings that are woven among the collagen strands of the tendon. And this is where the musculotendinous junction, where the muscle meets the tendon. These GTOs are very sensitive to slight changes in the tension of the tendon. They respond to both active contraction and passive stretch. Their information is carried on the type 1B fibers, which are fast. So, of course, this job is protective in nature, right? Their job is to monitor the tension in the muscle, to inhibit the contracting muscle or facilitate tension in an antagonist as a protective measure, so we don't cause damage to the muscle. So, these Golgi tendon organs are similar to the Ruffini corpuscles in the way that the dendrites branch through and weave through the collagen fibers of the tendon. Information is transmitted from the Golgi tendon organ into the spinal cord by those type 1B fibers. So other joint receptors. So again, the joint receptors respond to mechanical deformation of the capsule and the ligaments. So when there's a mechanical change in the shape due to mechanical deformation of the capsule and ligaments, these receptors are picking up on that. So there are ligament receptors. With this picture on the right is that red one. These are receptors that we find in the ligaments that connect muscle to muscle. They signal tension like the Golgi tendon organ. Tension in the ligament as a protective. That's what ligaments do, right? Try to give us some protection and some stability. So this information is carried on the 1B fibers as well. We also see free nerve endings throughout the joint capsule and in the tendons. They respond to the pain of inflammation. This information is carried on the type A delta fibers and the C fibers. We also see the Ruffini endings here. They are in joint capsules and ligaments. They respond to extremes of joint range. So if you imagine that's the ACL in the front of the knee, they're going to respond when you've got that leg and that knee in full range of motion. They're going to respond at the extremes of the joint range and respond to more passive they respond to more passive movements than active movements. So like when you're arranging a person or stretching a person, these Ruffini endings would be more active. This information is carried on the type 2 fibers, which are fast. And we also see paciniform corpuscles here in the joint capsule. They monitor rapid joint movements. They are silent when the joint position is constant. So again, we have a lot of different players when we talk about normal joint proprioception. Normal joint proprioception requires muscle spindles, joint receptors, and cutaneous mechanoreceptors. So that's a lot of redundancy in the system, which really tells us how important proprioception is to controlling movement, because there's a lot of overlap. So if part of that system is failing, let's say you've had a total hip replacement, we see with those folks, even with the loss of some joint proprioceptors, they continue to have good proprioception because of the overlap and the redundancy in this system. So sensory information, all of the pathways that carry sensory information are similar. The nerve impulses are generated from the original stimuli. The stimulus brings that sensory receptor to threshold at the trigger zone. The potential is propagated toward the central nervous system along the peripheral axon to the cell body or the soma in the dorsal root ganglion. Then that action potential travels into the spinal cord along that proximal or the central axon. And then once in the spinal cord, the information ascends via axons in the white matter to the cerebral cortex for further interpretation. So again, whether the receptors are bringing mechanical information or chemical information or thermal stimulation information, once that receptor potential exceeds the threshold of the trigger zone, an action potential is generated, and then it's conducted and heading into the central nervous system, and then it gets to the brain, and the brain kind of figures out exactly what that incoming information is and how and if we need to respond to it. So there are a couple different pathways that sensory information reaches the brain. We have these conscious relay pathways. Of course, proprioception, some proprioception is conscious. So we have information that is conscious, including touch, conscious proprioception, pain, temperature. We also have divergent pathways where the information is carried to many areas of the brainstem and cerebrum. Things like aching pain, it's a little more vague. We also have unconscious pathways. We have the cerebellum involved in proprioception, but it's not conscious proprioception. But the cerebellum has huge impact on our ability to make rapid automatic adjustments in our movement patterns and to maintain balance. So again, we have a lot of different ways that this information gets into the brain. And again, you've seen this before. I just put it in here because I wanted you to see the connection again. I wanted it to be closer to these next pictures. Again, the first order, the second order, and those third order neurons. All those three come together to form these pathways. And you've seen that before. I just stuck it in here because I thought it was helpful to see next to these upcoming slides. And again, these pathways carry sensory information from the skin and the muscles, the body wall, the head, the neck, and the limbs, and they are carried through three major somatic sensory pathways: the spinothalamic pathway, the posterior column or the dorsal column pathway, and the spinocerebellar pathway. Each pathway has pairs of spinal tracts, has symmetrically arranged on opposite sides of the spinal cord, and all axons within that tract share a common origin and destination.